In this video, we are going to learn about laminin. Laminins are a family of extracellular glycoproteins that consist of three different polypeptide chains linked by disulfide bonds and organized into a molecule resembling a cross with three short arms and one long arm. At least 15 different laminins have been identified before moving on to the complicated topics. First of all, let's learn about laminins, and which are basically consist of three different polypeptide, three different polypeptide chains linked by disulfide bond. Disulfide bonds are connecting these three different polypeptide chains, which resembles a structure with three short arms. Three short arms will be there and one long arm will be there. Okay, three short arms will be there and one long arm will be there. At least 15 different laminins have been identified. Like fibronectin, extracellular laminins can greatly influence a cell's potential for migration, growth, and differentiation. For example, laminins play a critical role in the migration of primordial germ cells. These cells arise in the yolk sac, which is located outside the embryo itself, and then migrate by way of the bloodstream and embryonic tissue to the developing gonad, where they eventually give rise to sperm or eggs. During their migration, the primordial germ cells transverse surfaces that are particularly rich in laminin. Primordial germ cells possess a cell surface protein that adheres strongly to one of the subunits of the laminin molecule. The influence of laminin on nerve outgrowth can be seen. In addition to which are the main points? First one was the laminins are consist of three different polypeptide chains linked by disulfide bonds. There are three short arms and one long arm. Until now, 15 different laminins have been identified, and this influence the cell's potential for migration, growth, and differentiation. Laminins play a critical role in the migration of primordial germ cells because they, move according to the transverse surfaces that are particularly rich in laminin. In addition to binding tightly to cell surface receptors, laminins can bind to other laminin molecules, to proteoglycans, and to other components of basement membrane also. The laminin and type 4 collagen molecules of basement membrane are thought to form separate but interconnected networks as depicted in the figure. These interwoven networks give basement membranes both strength and flexibility. In fact, basement membranes fail to form in mouse embryos that are unable to synthesize laminin, cause death of the embryo around the time of implantation. What's happening? The laminin and type 4 collagen molecules of basement membrane are thought to form separate but interconnected networks. These interwoven networks give basement membrane both strength and flexibility. In fact, basement membranes fail to form in mouse embryos that are unable to synthesize laminin. There won't be basement membrane, so there will be difficulty in this implantation process. So they won't develop. Mouse embryos don't develop. Here we can see a picture of a model of a basement membrane scaffold. Basement membrane contains two network forming molecules, collagen 4 and laminin. There will be thickened cross shaped molecules. The collagen and laminin network are connected by entactin molecules. Entactin molecules. After this, let's learn about one more topic, that is nothing else but the dynamic properties of the extracellular matrix. The micrographs and diagrams presented in the first section of this chapter portray the ECM as a static structure. In actual fact, however, the ECM can exhibit dynamic properties both in space and over time. Spatially, for example, ACM fibrils can be seen to stretch several times their normal length as they are pulled on by cells and contract when tension is relieved. Temporarily, the components of an ECM are subject to continual degradation and reconstruction. This process serves to renew the matrix and to allow it to be remodeled during embryonic development or following tissue injury. Even the calcified matrix of our bones, which we think of as a stable inert structure, is subject to continual restoration. The degradation of extracellular materials, along with cell surface proteins, is accomplished largely by a family of zinc-containing enzymes called matrix metalloproteinases. Matrix metalloproteinases (MMPs), matrix metalloproteinases, which are associated with degradation of the extracellular materials. Matrix metalloproteinases that are either secreted into the extracellular space or anchored to the plasma membrane. As a group, MMPs or matrix metalloproteinases can digest nearly all of the diverse ECM components, although individual family members are limited as to the types of extracellular molecules they can attack. As a group, MMPs can digest nearly all of the diverse ECM components, although individual family members are limited as to the types of extracellular molecules they attack. The physiological roles of MMPs are not well understood, but they are thought to be involved in tissue remodeling, embryonic cell migration, wound healing, and the formation of blood vessels. Uh, as might be expected from enzymes whose normal function is to destroy extracellular materials, the excessive or inappropriate activity of MMPs is likely to cause disease. In fact, matrix metalloproteinases have been implicated in a number of pathological conditions, including arthritis, tooth and gum disease, formation of blood clots and heart attack, and tumor progression. At least three inherited skeletal disorders have been traced to mutations in MMP genes. MMP in the sense matrix metalloproteinases, which is related with the degradation of extracellular materials. Okay, so